Hello everyone and welcome to today's live broadcast, Prediction of Enzalutamide Sensitivity in Triple-Negative Breast Cancer, presented by Dr. Joel Parker. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's presentation is sponsored by NanoString Technologies. NanoString Technologies is a publicly held provider of life science tools for translational research and molecular diagnostics. The company's technology enables a wide variety of basic research, translational medicine, and in vitro diagnostics applications. NanoString's products are based on a novel digital molecular barcoding technology invented at the Institute of Systems Biology in Seattle under the direction of Dr. Leroy Hood. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social network website provider of virtual events and webinars advancing scientific collaboration and learning. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you, so questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of your slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button at your lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Joel Parker. Dr. Parker's research is focused on the methodological development and integrated analysis of high-throughput genetic and genomic studies. He previously led the development of algorithms and content resulting in Procigna TM and only CE marked and the FDA 510K cleared breast cancer diagnostic assay for FFPE tissue. He is currently involved in similar diagnostic development in kidney and bladder cancer and recently presented a novel predictive biomarker for the enzalutamide in triple-negative breast cancer, which is being used as entry criteria for a phase 3 trial. Dr. Parker currently directs the sequencing, microarray, and other genomic analysis in the bioinformatics shared resource at the Lineberger Comprehensive Cancer Center. The impact of his collaborative efforts are demonstrated in publications which are presently constitute over 150 peer-reviewed publications. I'd like to now turn over the presentation to Dr. Parker. Welcome, sir.
Thank you very much, and thanks for that nice introduction. So today I'll get to talk about enzalutamide and our development of this biomarker. I did this work with a company called Medivation, who produces Xtandi, who's also going by enzalutamide. And this is in collaboration with Katrina Carey and Hardesh Upal. So enzalutamide is a potent inhibitor of AR signaling. Androgen receptor is one of the key components in prostate cancer and is used, and this drug is used in metastatic castration-resistant prostate cancer, where it gives a significant benefit in overall survival. In this particular study, we wanted to look at its activity in breast cancer. But what is known is that it actually inhibits AR signaling in multiple different ways. It inhibits binding of androgens to AR. It inhibits the nuclear translocation and also inhibits androgen-mediated DNA binding. So in this phase 2 study of enzalutamide here in triple-negative breast cancer, women were recruited if they were androgen receptor positive, meaning that they had some staining of androgen receptor by immunohistochemistry, and that was basically any staining. So it was a very sensitive assay. And they had to have advanced triple-negative breast cancer, and triple-negative being defined as lack of staining for estrogen receptor, for HER2, or for progesterone receptor. They could have any number of prior therapies and allowed for bone-only disease, but no brain metastases were allowed. Also, these patients were, when assayed tissue was taken for for genomics, as you'll see in just a minute. The primary endpoint of the study was clinical benefit rate at 16 weeks, which had to be used due to the aggressiveness of these metastatic triple-negative breast cancers. We also collected other key endpoints such as 24-week clinical benefit, progression-free survival, and overall survival, which I'll present today. This was a single-arm or Simon's design study where in stage 1 we had to see at least 3 of 26 evaluable patients have a clinical benefit at 16 weeks. And if so, progressed to stage 2, which the study did. And in that subset, see greater than a total of greater than 9 out of 62 patients to constitute rejection of the null hypothesis that there is clear benefit of androgen receptor. So the interesting thing about this drug in this study is that the triple-negative breast cancer is typically treated with cytotoxic chemotherapy. And cytotoxic chemotherapy, as we all know, has many very bad side effects. In contrast, this drug enzalutamide has a very conservative safety profile in that it only has grade 3 adverse effects of fatigue in a very low percentage of patients. So it provides a much better quality of life for the patient. And in this study, this was single agent. So these women received no cytotoxic chemotherapy. They only received this hormone therapy as part of the study. One of the initial hopes was that IHC would be able to understand, it would be able for us to evaluate and identify those women that would benefit from the drug, as androgen receptor, the drug target, can be easily stained for. And what we see in these plots, first on the left in the intent to treat population, is that staining at a threshold of greater than 10% produces some but still modest benefit, with progression-free survival extending from 8 weeks to 14.5 weeks of 50% or 50% survival. If you reduce the population to just those women who received 0 or 1 prior therapy, so they weren't extensively treated prior to entering the study, that benefit is increased, with 50% surviving at 8.5 weeks when they had less than 10% AR staining, and 17.6 weeks for those that had greater than or equal to 10% AR staining. However, this was not statistically significant, and the samples were used further for biomarker analysis. So this slide demonstrates the kind of the comprehensive set of biomarkers that were assayed as part of the study. RNA sequencing was used for to assay the whole transcriptome. This was using a ribo-0 technique to remove all the ribosomal material and then sequence total RNA after that. This led to the evaluation by PAM50, a classification technique commercialized as the Prosigna diagnostic, which provides information about individual subtypes of breast cancer and is indicated for producing risk of recurrent scores. Other classification procedures that we investigated include the Layman classification, which is specific for triple-negative breast cancers, and notably includes an androgen receptor high subgroup or androgen-sensitive subtype potentially. And then we also use this to mine for novel signatures of androgen receptor gene sensitivity or drug sensitivity. And then there was also DNA available, and this DNA was utilized in order to look for mutations that might be useful in determining those patients who were likely to be sensitive to the drug. Now, in all these cases, development has to occur. However, the amount of development to get through to a successful test varies, in that with things like the PAM50 classification, because it's already commercialized, that it's possible that simply using those same subclasses or subtypes that are defined by the PAM50, if they are predictive, would allow for a very rapid translation to the clinic. In contrast, these other biomarkers or candidate biomarkers would have to go through extensive development in order to see the light of day as a clinically useful classifier. Prior prior to going deep into biomarker development, we first wanted to understand were there associations with response. What we're looking at here is a heat map of indicating the gene expression of a few hundred genes, 521 genes, that were significantly associated with enzalutamide response. The color bar on the top indicates those that are AR positive by IHC in the black, and then those in gray at that top color bar were AR IHC negative, and thus were not part of this intent to treat population. The second color bar shows the PAM50 subtype, and I'll just point out that red in that color bar is the basal-like subtype, while other colors represent various luminal subtypes, luminal A, B, HER2, and the normal-like. The third row shows blue and gray, where gray represents those patients that did not achieve 16-week clinical benefit, and blue are those that had a response or had clinical benefit at 16 weeks. When clustering over this set of genes, where red indicates high expression and green indicates relatively lower expression, we see a cluster on the left. Get my marker up. That shows very high expression of a set of genes, including the androgen receptor. This particular cluster is also enriched with responders, shown in blue, and is typically non-basal-like by the PAM50 subclass. On the right-hand side, we see the group of samples that are almost 100% basal-like, and there are very few responders in this group, as noted by almost no blue in the gray versus blue color bar, and a very different gene expression profile, with the majority of genes being highly expressed, the majority of these 521 genes being highly expressed in that basal-like group. When we did enrichment analysis for these two groups of genes, that is those high in the non-basal group and that are responsive, and those high in the basal-like group that don't typically respond, we find biological meaning in that we find breast cancer subtypes being enriched in both of these classes, notably that the ones on the right being basal-like and those on the left, high on the left side of the heat map, being more luminal-like. We also find signatures of apocrine response from a paper by Farmer et al. That I'll get to in a moment. And also, I don't know if this is listed here, but we also saw signatures of androgen receptor activity from cell line-based models. When we limit this analysis down to just those that are basal-like to try to produce the most homogeneous population that we can, we remove many of the responsive cases. In fact, only 6 or 7 responders show up within this group of basal-like cases. However, when we test within this group of only basal-likes, what we see is a similar expression profile, showing that even those that are called basal-like but do respond have signals that are relevant to basically non-basal or more luminal-like disease. And that's shown by these gene sets that pop up in the non-responder patients. That's these patients here in blue, showing targets like of differentiation, which is typically with this particular gene set goes along with proliferation and others. And then on this next slide, we dig a little bit more deeply into the PAM50 measures. And we're looking here at the distance, instead of just calling them specific subtypes of basal versus luminal. We're looking at individual subtypes, or the quantitative measure of these individual subtypes in order to see how strong these different measures are. And what we find is that the distance to basal, shown in the box plots on the top left, strongly segregates responders from non-responders. With those patients that are very close or very similar to basal-likes being non-responders, and those that are more distant having a negative correlation being, did I say that properly? Those that are more similar to basals having a strong positive correlation being non-responders, and those that are far from basals or having a large negative correlation being responders. While HER2 measures to the left of that, or to the right of that, don't show much difference, and then luminal A measures show the contrast of the basals, with those that are being more different than luminal A's being non-responders, and those being more similar to luminal A's, showing larger positive correlation, typically being responsive. And when we just plot these two measures against each other, basically the correlation to luminal A versus the correlation to basal in the scatter plot on the far right, you see that these two measures are strongly correlated with a negative direction. And what we find is that when we color by non-responder versus responder, that those patients that are typically stronger correlates to luminal A and negatively correlated with basal are enriched in the responders. And this is the signal that we'll continue to exploit as we develop our biomarker. But before we look further at the PAM50 calls, we wanted to spend some time with the apocrine subtype. This subtype was first described by Farmer et al. In an Oncogene paper in 2005. In this paper, they looked across a number of different public data sets listed here and showed that in every one of them there is a small but definitely present set of patients which display this apocrine or androgen receptor driven like phenotype. So we wanted to develop a predictor of this particular phenotype, and using the Farmer data set, which is shown on the right, we tried to look for a set of genes that would be predictive of this apocrine subtype. In the heat map on the right, the basals are colored with red in the color bar at the top. Those that were apocrines from the paper were colored in purple, and those that were luminal are colored blue. We used a simple linear modeling based strategy in order to determine a set of genes that were predictive of these three classes. And what's shown on the left is the accuracy of that classifier across cross-validation test sets relative to the number of genes that were used in the model. And so in the end, we used about 200 genes, which produced around a 94% accuracy in those genes are shown in that heat map on the right. And at this level, we could produce a classifier that we could take over to our study cohort and predict apocrine subtype and understand if that particular subtype, those predictions were associated with response. So here's that result. On the left are the 200 genes from the visualized in the Medivation cohort or the enzalutamide study. The top color bar is showing predictions based on the model built from the Farmer et al. Data. The predictions in purple are the apocrines, the blues are luminals, and red predictions are basals. In the second color bar, the gray and blue, again the gray are non-responders and the blue are responders. And what we see is that the apocrines in purple are enriched for those blue or responsive samples, whereas the basals on the right-hand side have a much lower number of responders, and this matches the general features that we see when looking at the PAM50 genes as well. Now, if we take this apocrine prediction to be our biomarker, we can see in the table in the middle that of the apocrine predictions, about 50% of them, 12 out of 24, are responders. However, in the basal and the luminal groups, while the responders are at a much lower frequency or much lower proportion, the actual numbers are still well above zero. So that if we took this predictor to be our biomarker, we would miss many of the positive cases or many of the responsive cases, despite showing certainly accuracy that's well above chance, as you can see through the sensitivity and specificity curves at the top right or the ROC curve on the bottom right. Another technique that's out there for classifying triple-negative breast cancer I alluded to earlier was from this Lehman paper. This was in JCI back in 2011. And in this paper, they nicely demonstrate that subtypes of triple-negative breast cancer can be reproducibly identified. They divided triple-negative breast cancer into two different basal-like categories, an immunomodulatory group, a mesenchymal-like group, a mesenchymal stem cell-like group, and the luminal AR group, which we are most interested in here. So this was done based on a larger panel of 3,247 genes. And our subtype classifications using layman were produced using the tool that was described in that paper. So here we can choose two different strategies for biomarker development. The diagnostic positive cases could simply be those that are luminal AR. However, as I'll show you in a second, it would be much more productive in order to just describe diagnostic positive cases as those that are not the basal 1 or basal-like 2 groups. And that's shown here as a bar plot. So what we see on the left-hand side are the basal-like 1 and basal-like 2 categories, and blue is showing the number of non-responders, with the green bars stacked on top are those responders. So in our diagnostic negative or basal-like classes, we get fairly large enrichment for non-responders, as we saw with the PAM50 or the apicrine, or the farmer, that is, based prediction strategies. Then our diagnostic positive subgroups, we see that there's enrichment, general enrichment of responders, and it's especially true in the luminal AR group, which points to this subtype generating the correct biology or biological association for this group, because they have the largest enrichment of responders to the drug. However, the high level of non-responders in this diagnostic positive population produces a low positive predictive value. That is, many samples that would be considered positive would not respond to the drug. Now, we could play with this and get rid of the mesenchymal group, which has none, move those over to the negative category, that is the basal-like category, and there's other things we can do here. But in the end, no good strategy was able to clearly segregate diagnostic positive from negative, as you can see by the way the bars are stacking here. So in our last attempt using known strategies, this is the PAM50-based classification technique that I discussed a little bit earlier. The 50 genes are shown on the left in a heat map, which demonstrates the profiles of the 4 tumor types of interest: luminal A, luminal B, HER2-enriched, and basal-like. And the way an individual sample is classified is shown in the middle, in that a single sample, which is unknown, is taken and compared to each one of the individual profiles, and is assigned the class of the profile that is most similar. So this would be assigned profile A here, likely, which looks more like the luminal A category. And in this case, we would take diagnostic negatives to be basal-like and diagnostic positives to be all those non-basal categories, similar as we did with layman, with the layman classifier. In this case, the PAM50 subtyping shows an enrichment of responders in the diagnostic positive category. However, there are plenty of responders in that diagnostic negative or basal-like category. And so while this would work in enriching for those that are positives, it would miss many patients who would respond to the drug, which is also a trade-off that we weren't quite willing to make at this level of the analysis. So we continued further and looked more in depth at positive predictive value, sensitivity, specificity, and negative predictive value of the different classification schemes. Here on the left is just revisiting the layman-based scheme versus the PAM50-based scheme on the right, and just contrasting the high level of non-responders in diagnostic positives for layman versus the high loss of responders in the diagnostic negative population for PAM50. So this motivated us to go through a model development exercise. As part of this exercise, we used a number of different feature sets, which is represented by each of the curves in these two charts on the left. So what's shown is our clinical measures reported out as clinical D or V. These are different IHC-based classifications using different antibodies, and each one of these clinical variables actually incorporates 12 different measurements of IHC, including things like for everything from total counts to nuclear counts versus cytoplasmic counts and their ratios and whatnot. We also included PAM50-based variables, which are the discrete subtype calls, as well as the distances or correlations to each centroid, as I showed you earlier. And we've included these features individually or combined, as well as different weighting schemes of the 50 genes that were in PAM50, the apricron-based predictor, which is shown by the green curve, and then all of these features together in a single model, which is the orange curve. These were all modeled using a Monte Carlo cross-validation approach, such that 2/3 of the training set was taken out, a model was built, and then that model was evaluated in the third that were held out as the test set. That entire procedure was repeated 500 times, and prior to that entire exercise, a validation set was set to the side, which we'll come back to. As part of this process, we wanted to see what feature set, as well as what number of predictors in that feature set, gave us the best overall accuracy. And on the top left, you see that many of these curves look very similar. So the general performance of any of these feature sets was quite similar, and that's a good thing. They're all honing in on the same biology. On the bottom left, you can see that if you have to pick a winner, that based on this Monte Carlo cross-validation analysis, the light blue curve with as few as 1 or possibly 2 features, and that is the PAM50-based scores, gives us the best general area under the curve. So to visualize that one model another way on the right, now named the PREDICT-AR biomarker, the resistant-based predictions show a huge enrichment of non-responders, whereas those that are predicted to be sensitive to drug have a relative enrichment in the responders shown in blue. Sensitivity, specificity, positive predictive value, and negative predictive value of this biomarker is shown at the top and this is estimated from the Monte Carlo cross-validation test sets. Importantly, the positive predictive value is 45%, meaning that 45% of patients predicted to be sensitive to the drug will actually be, while 90% of those patients in the NPV case that are not predicted to be sensitive would not benefit from the drug. And this is in contrast to IHC, estimated, where we've estimated these measures the same way. And just to close the book on IHC in this particular biomarker case, we see a 10% drop in PPV and a similar drop in NPV, showing clear improvement of the PREDICT-AR signature relative to IHC. So at this point, we've developed a diagnostic assay for enzalutamide in triple-negative breast cancer. The patient's primary tumor, or FFP sample, is run through this next-generation sequencing assay. 50 genes are extracted. These are the PAM50 genes. We measure the distance to each of the centroids or each of the subtypes, and using the distance primarily to luminal A and then secondarily to basal-like, these two measures are combined to form our model of enzalutamide sensitivity. Evaluating the data in different ways now from the same cohort, we look at first waterfall plots of time on study. And what you see on the left are the PREDICT-AR negative patients, which generally have shorter times on study, and with only a couple of patients which are still active, indicated by the orange arrows. On the positive, PREDICT-AR positive cases, you can see that many patients are still on study, still active on study, taking the drug, as well as, in general, the duration on study was much longer. If we look at progression-free survival, again, the model was not trained on progression-free survival. It was trained on the 16-week clinical benefit. And what we see is that in the intent-to-treat population, that our median progression-free survival is improved from 8 weeks to 16.1 weeks for the entire population. And if we limit to just those where they're getting this hormone therapy instead of cytotoxic chemotherapy as their first or second line of therapy after disease progression, the median survival goes from 8.9 weeks to 40.4 weeks. So this is a tremendous increase in progression-free survival for these patients. In a multivariate analysis, utilizing both the PREDICT-AR model as well as age and IHC, we see again that the PREDICT-AR model provides significant predictive value with the hazard ratio of around 0.45, second only to number of prior therapies, with 0 or 1 prior therapies being beneficial in this cohort. And this effect carries through to overall survival. So what we're looking at here is overall survival from the cohort broken again by PredictAR status, with PredictAR negative patients, 50% of them surviving out to 32 weeks, and while the PredictAR positive patients surviving out to 75 weeks, likely with a much better quality of life in addition to this improved survival time due to the nature of the drug. Now, it's possible that this, because we know that the model is weighted towards being luminals or at least not basals, and those patients will do better in the absence of any drug, we wanted to ensure that this particular signature was not simply prognostic, but it was predictive, because this was a single-arm trial with no placebo-controlled arm. Our best evaluation of this was making use of this neoadjuvant data set published in 2011. We extracted triple-negative portion of this data set and applied the PredictAR signature to it, and we'd see no difference in distant relapse-free survival according to diagnostic arm in these patients that did not receive the drug, which indicates that the biomarker is likely predictive and hopefully not simply prognostic. Now, at this point, this is typically where research has to end, and thus this is then turned over for commercial development. But a lot of times that's where it fails, because it's very difficult to translate one of these technologies into a meaningful and accurate and reproducible technology that can be clinically utilized. Now, what's different here is that we have this wonderful tool from NanoString called Prosigna, which is already on the market and allows for classification and measurement of gene expression from FFP tissues, and has already been validated in breast cancer. In this triple-negative cohort, because we see that our biomarker can make use, and actually, the best use of the biomarkers we tested was based on the same set of genes that are assayed by Prosigna, that is the PAM50 genes. So we hopefully had a quick means of translation to the clinic, and that's because a lot of the work that needs to be performed in order to make that translation happen had already happened for the Prosigna assay. But I want to visit that now just so it's understood the level of effort that it takes in order to move one of these assays into clinical utilization. So the Prosigna assay is already a validated platform, has more than 300 systems worldwide for testing breast cancer and producing the risk of relapse score. It was based on a paper in Journal of Clinical Oncology in 2009, and the model captures the heterogeneity of breast cancer gene expression in producing the model, so in producing the prognostic outcome. So it's not simply trained directly on the outcome without knowledge of the underlying biological heterogeneity. And in developing this assay, a number of very in-depth studies were performed on behalf of NanoString and in collaboration with myself and other academics in order to ensure repeatability and accuracy of this assay. The first such set of experiments were described in this paper by Nielsen. In this RNA precision study, there were greater than 100 assays used to assess variation. So there were a set of 10 samples. These were basically one for each subtype: basal, HER2-enriched, luminal A, and luminal B. I guess 2 forms of luminal B here. And then there were 2 examples of each of these, so 10 independent samples. And for each of these 10 independent samples, they were run through different sites and at different operators at each site, different reagent lots by each operator, and different runs for each reagent. So 4 levels of replication here to produce a very large set of data that we could use to analyze repeatability and accuracy at multiple levels. What we find and report in this paper is that the ROR scores across all these levels of all these levels of variation were extraordinarily tight, extraordinarily accurate, and very reproducible. And you can see that in the top left by the low variation in the ROR scores, which is expanded out into histograms in the rest of these for each individual sample in the study. So the ROR only varied by a couple of points for each individual sample across all these levels of replication. And variance component analysis indicated the predominant variation was within run and then reagent lot. Another experiment that was performed was to take a larger pool of samples and assay them at 3 independent sites with independent pathologists, operators, RNA isolation kit lots, and then run in duplicate. And so for each of these, we're seeing on the right the resulting risk of relapse score relative to the tissue average on the x-axis. So there should be stacks of points representing each individual sample, and you can see that within each kind of vertical stack of points, there's very little variation, only a couple of ROR units across these 3 completely independent runs of this very large sample set. We also found that in other studies that some amount of normal contamination could produce variability. This was initially published by Fadi Alumi and Melissa Troster back in 2011. And we looked at its effect on ROR relative to the percent non-tumor tissue more recently in this study in 2014, and find that the type of contamination does play a role, and thus pathologist review is necessitated and required prior to putting these studies through the assay, these samples through the assay. The result of all this work is the Prosigna test, which is an amplification-free and automated protocol that can turn around samples in 2 to 3 days. It has 510K FDA clearance and is CE marked for FFPE tissue, and produces the reports like you see here to generate the risk of recurrent scores. This assay we could then leverage, because of all this development, we could then leverage this as much as we can in order to get the test that we're talking about now, which is this enzalutamide sensitivity test, to market as fast as we can. And what I've shown you then is that a lot of steps are required. Characterization, verification, validation, all these things would be required in order to take any of the other assays that might be viable and could be perfectly accurate biologically, but to make them clinically actionable. But because we have this test in place and we can produce the proper measures in order to provide sensitivity measures to enzalutamide using current instrumentation, a study was designed in order to test this in a more robust manner. In this randomized, double-blinded phase 3 study called the ENDEAR, patients will be screened using the assay discussed today, and those that are considered diagnostic positive will be randomized to one of 3 arms. An enzalutamide plus paclitaxel arm, which will be compared against a placebo plus paclitaxel arm to understand the improvement in this diagnostic positive population. A third group will also be assayed, which will be the single agent arm to match the phase 2 trial. And this particular trial will be run worldwide, hoping to enroll approximately 2,200 or screen 2,200 patients to enroll around 780. And with that, I'd just like to stop and thank all the folks that were involved in development of this diagnostic. First, the PAM50 commercialization to Procigna, and all the work that had to be performed in order to get that done. That would include the folks at the Huntsman Cancer Institute, like Phil Bernard, Torsten at UBC, and then, of course, our colleagues at NanoString, and where I am, and with Chuck Peru, as well as others here at UNC. Then I'd also like to thank those at Medivation, such as Herdesh and Amy and Katrina, for helping us push through with this triple-negative study of enzalutamide, as I believe it's going to be very beneficial to patients that are diagnostic positive. And thank you all for listening.
Thank you so much for your presentation, Dr. Parker. We want to start our Q&A segment. So here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If we are unable to get to your questions due to time constraints, Dr. Parker will be answering submitted questions via email. So let's begin with our Q&A. Our first question, Dr. Parker, is how can you be sure the signature is predictive of sensitivity and not just simply prognostic?
So, yeah, that's a tough one because it wasn't a randomized study for this phase 2. And so ultimately the only way we could know if it is truly predictive is to have a placebo-controlled phase 2 study. Because of the nature of this study being single arm, we only have kind of one side or one way to look at this data, and that's in those patients that receive the therapy. What we'd like to know is in a randomized trial, is this signature also not providing benefit in those that did not receive drug? As I showed you, the best estimate we have of that comes from public data, which is the study by Hatsis et al. Back in 2011. In that set, they were treated with a standard chemotherapeutic regimen and followed up so that we could look at disease-free survival estimates. Those data were microarray data, so they were a completely different protocol or assay protocol than anything else I showed today. So that has to be taken into account when evaluating it. However, I do feel that that data set is a very good data set in both representation of the population as well as the quality of the assay data that was produced. And thus I'm confident in those estimates and in our ability to subtype those samples. And I'm confident in the diagnostic positive negative classes that were assigned. And in that study there was no difference. So that's our best estimate so far. However, it will never take the place of having a true control arm as part of the clinical trial.
Thank you for that. One audience member would like to know, will you be able to run this test with RNA sequencing? So
this question is, it's interesting because we did develop the biomarker with RNA sequencing. So that seems very reasonable, and a lot of institutes now have RNA sequencing capability. However, I hesitate to recommend it at all because of the nuances of RNA sequencing. I love RNA sequencing. I'm so glad. It's been such a wonderful tool for discovery, and made this study possible by being able to discover the biomarker using RNA sequencing. However, there's so many RNA sequencing protocols out there, and many of them are not clinically viable simply because they won't work well on FFP tissue. And when you whittle it down to just those protocols that do, they have just not been. They may well have very good accuracy and repeatability. Of course, I believe they do, but it just hasn't been shown at the level needed yet in order to use it as a clinical tool. So while one day it may be possible, right now, and the reason we look to NanoString to do this is because we believe these data can help people right now. And so we want an assay that we don't have to wait and develop. NanoString gave us that ability by having the ability to run RNA expression estimates from FFP tissue and having devices in the market that can do that right now.
Thank you. Thank you for that important response. We have some great questions coming in, but we only have time for one more. An audience member would like to know, does the model provide any biological insight into the disease?
So, let me think about it. So my short answer is yes, but that's, there's certainly some room for interpretation here, because we haven't done anything functional yet to show this, but my thoughts here are that just simply the context of this study being triple-negative breast cancer, that means they lack estrogen receptor, they lack progesterone receptor, and they lack HER2. Yet the model is picking up on those patients that of the triple-negative population look like luminal patients, which are typically ER positive, PR positive, and sometimes HER2 positive. And so my thought is that androgen receptor positive or androgen receptor sensitive patients are these patients where androgen receptor is somehow taking the place of estrogen receptor or progesterone receptor in driving the disease. Because the signaling that we see, or at least the gene expression patterns that we see, look like luminal tumors where typically estrogen, progesterone, HER2 are driving the disease. Here what we see is the lack of those markers, and those are the samples that are sensitive to androgen receptor. And so just putting those things together, I feel like what the model is telling us is that since it's the luminal tumors that are showing sensitivity, that androgen may be driving some subset of triple-negative tumors to really be of the luminal subtype.
Thank you for that, Dr. Parker. Do you have any final comments that you'd like to share with our inquisitive audience today?
Just thanks for letting me present this work. If it didn't come through, I'm extraordinarily excited about it. I think for me, this is, a lot of people talk about the promise of genomics, and this was one instantiation of it, because in this instance, we take a drug that's been approved in a completely different disease, and we identify patients which are using genomics, which should be, a subset of patients in a disease which the drug is not indicated, that should be sensitive, and by doing so, take a drug that has, a fairly good safety profile, apply it to the right subset of patients in a different disease, and the result is a doubling of their overall survival time with potentially better quality of life during that time. And so personally, I hope we can just continue to see the fruits of genomics by applying it to clinical trial scenarios like this. So far, genomics has told us a lot about biology and has been used as a wonderful tool in basic and some translational research. What we're seeing here is really the power of it when we can apply it in the context of clinical trials. And I really hope this spurs more use of genomics in true clinical trials like this one.
Thank you so much, Dr. Parker. I'd also like to thank today LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through July 2017. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay, and we encourage you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you next time. Goodbye, everyone.